Okay, in this section, we are going to start talking about what we call the endomembrane system. As you might imagine, this is a system of membranes. Endo means within, so this is within the cell. Get it? The words, they start to make sense after a while. These are going to be a group of structures that make things and sometimes break things, but put things in a shape that the cell is going to need. So I'm going to call these the factories of the city, or the workhorses of the office. So the endomembrane system, like I said, it's a group of structures. These are all organelles, so they are all surrounded by membranes. Those are phospholipid bilayers. They're all going to work together. They each have a different job, but they're going to work together to make a bunch of different kinds of things and then transport those things, put them in a form that can be transported around the cell. So the endomembrane system includes the smooth endoplasmic reticulum and the rough endoplasmic reticulum, the Golgi apparatus, the nuclear envelope, which we already talked about, lysosomes, and vesicles. So we're going to talk about all of these things. Now, the endoplasmic reticulum is actually made up of two parts. They're usually connected to each other. And in most of our diagrams and our models, when they're colored, these are always the same color. So use that as a hint when you're looking at a different diagram or when you're looking at a model in the classroom. The endoplasmic reticulum, the rough and the smooth, are both usually the same color. Now, the rough endoplasmic reticulum is called rough because it has a rough surface. So it's the one that's bumpy. So this part that has these little blue bumps on it, that's the rough endoplasmic reticulum. The smooth endoplasmic reticulum is this part over here that's smooth. It doesn't have bumps on it. Is it that simple? Well, yeah, actually. You do have to know how to spell endoplasmic reticulum, but the smooth rough part is pretty simple. Okay, endoplasmic, it's within the cytoplasm. Reticulum refers to a network or a lattice or interconnected things. So this is basically a system of membranes that are connected to each other. All right, so two different functions, two different types of functions. The smooth endoplasmic reticulum, its main job is that it is going to synthesize and store lipids. Those are fats. Phospholipid bilayer, for example, is going to be made by the smooth endoplasmic reticulum. It's also going to break down sugars, and it is going to detoxify things that could be toxic, poisons, but also drugs and alcohol. So one of the things about developing new medicines is to get them past the smooth endoplasmic reticulum so that the smooth endoplasmic reticulum doesn't think they're toxic and need to be broken down. That's actually one of the challenges of making medicines. So smooth endoplasmic reticulum makes lipids, breaks down sugars, and detoxifies toxins. The rough endoplasmic reticulum is going to make proteins, and that's why it has bumps. The bumps are ribosomes. Those proteins that are made by the rough endoplasmic reticulum, those are going to be used either in another organelle, or those proteins are going to go to the plasma membrane. So all of those proteins that we were showing embedded in the plasma membrane, those were all made by the rough endoplasmic reticulum. Now, the rough ER, and yes, you may call it the rough ER when you were talking about it, but you will write out endoplasmic reticulum on the test. The rough ER does do some modification, folding the proteins and making sure they're in the right shape, but its primary role is synthesis, making the proteins, okay? And that's why it's covered with ribosomes. Remember, ribosomes make proteins. Now, I do want to mention there are two types of ribosomes in the cell. And I should have just used this diagram for this. These are bigger. These ribosomes on the rough endoplasmic reticulum, these are called fixed ribosomes. They are going to make proteins that are used on a membrane or inside a structure bound by a membrane. So in another organelle, such as the smooth endoplasmic reticulum, the proteins inside the smooth endoplasmic reticulum that are making lipids, those proteins were made on the rough endoplasmic reticulum. Now, the free ribosomes are just floating around in the cytoplasm. They are not inside a membrane. Remember, they're inclusions. They're not in a membrane. They are going to make the proteins that are in the cytoplasm that are not inside a membrane. So all of these little proteins that help the cell to keep its shape, that act as roadways to move things around within the cell, those proteins are made by free ribosomes. Okay, free ribosomes are inclusions. They are not organelles, and they are not part of the endomembrane system. I just wanted to mention them here because we were talking about the fixed ribosomes on the rough endoplasmic reticulum. Okay, now back to the endomembrane system. Next up is the Golgi apparatus. You may have heard it called the Golgi body or the Golgi complex. All of those names are fine, but do capitalize Golgi. This is named after the scientist who discovered it. This is kind of like the shipping, the processing center, the UPS store of the cell. So when the smooth and the rough endoplasmic reticulum make new molecules, they're going to put them inside little bubbles surrounded by a membrane that are called vesicles. A vesicle is a little structure with a phospholipid bilayer around it, and then something inside. And these are very small, and they're used to move things around within the cell. So the rough endoplasmic reticulum, it's going to make proteins, put them in a vesicle. Smooth endoplasmic reticulum, it's going to make fats, put them in a vesicle. Those vesicles are then going to go to the Golgi apparatus. The Golgi apparatus is going to take that vesicle, open it up, see what's inside, modify it if it needs to. If the protein needs to be refolded, if it needs to be cut up into smaller pieces, if the lipid needs something attached to it, the Golgi is going to do that. Then it's going to make a new vesicle with the proteins and or lipids in it, and it might combine proteins from several different vesicles into one. And then it's actually going to put a label on it, some kind of molecule that can be recognized by another part of the cell. And then it's going to release that vesicle, and that vesicle is going to be carried somewhere else in the cell. Now, that vesicle can either go to another organelle, or it can go to the plasma membrane, fuse with the plasma membrane, and either release those proteins or lipids to the outside of the cell, or if the proteins are embedded in the membrane of the vesicle, then those proteins become embedded in the plasma membrane, because the vesicle is just going to fuse with the plasma membrane and become part of it, because it's all phospholipid bilayers. Is that kind of cool? Let me show you a picture. Okay, so here's the nucleus up at the top of this image. All of this blue stuff, this is rough endoplasmic reticulum. We know it's rough because it has dots on it. It has a rough appearance. Those dots are ribosomes. They're making proteins. That is going to, those proteins are going to be put in a little vesicle. Here, this little blue blob, this little blue circle is the vesicle. That's going to go to the Golgi apparatus. Now, the Golgi apparatus has two sides. One is either called the receiving side or the cis face. The other is the shipping side or the trans face. Cis just means on the same side. Trans just means on the other side. So this cis face is on the same side as the rough endoplasmic reticulum, and the trans face is on the other side. That's all cis and trans mean. And so this is going to be processed, move through these different layers of the Golgi apparatus, and then the Golgi puts it in a new vesicle. And this one is going to fuse with the plasma membrane. So you can see this green part is the original plasma membrane, and then this little orange part is the vesicle. And they fuse together and they open up a hole, and then the protein that was inside, this one was in the membrane of the vesicle, and now it can be part of the plasma membrane of the cell. And that's how all of those trans membrane proteins get into the membrane. I think it's kind of fun. One of the other things that the Golgi apparatus can make is a lysosome. And a lysosome is a larger structure. It's actually an organelle in and of itself. And it has a bunch of different proteins in it. And all of those proteins are what we call enzymes. In this case, these are enzymes that are going to break down big molecules into smaller molecules. That way, the cell can recycle things that it doesn't need anymore. So what we have here in this image, we have a little lysosome over here. These little blue dots in it are proteins. Those are the enzymes. And then we have another vesicle containing, in this case, a damaged organelle. It could also be something that was engulfed by the cell as a way of the cell breaking down something from the outside. The lysosome fuses with the vesicle, and then the enzymes basically eat away and digest the thing that is now inside the lysosome, and that breaks it down, and then the cell can recycle all of the stuff. These are used in damaged cells. If a cell is damaged, but the whole cell isn't dead, the lysosomes can fuse with the damaged thing, break it down so that the cell can make a new one. Or if a cell itself is damaged to the point that it needs to be destroyed, the lysosomes will actually control that process. Also, some cells are just no longer needed, and they get broken down by their own lysosomes in a very controlled method that doesn't damage the cells around them. So one of the ways this happens is during embryonic development, the fingers and toes actually start out fused together, and then the bones grow, and then the tissue between the fingers and toes goes away. And that's lysosomes doing that, lysosomes within the cells in that tissue, breaking down those cells so that the fused part can go away. And that's why you have separate fingers. Yeah. Now, another type of cell that has a lot of lysosomes, oop, what happened to you? There you go, are some of your white blood cells, particular neutrophils. So I'm going to show you this video. And this is going to show a neutrophil. And I always have to wait for the burp. So this cell, this neutrophil cell, I don't want that. Okay. The neutrophil cell has a bunch of lysosomes inside of it, so that after it catches the bacteria and engulfs it and brings it inside itself, this neutrophil can then fuse that bacteria with the lysosome and then break down the lysosome, or break down the bacteria, sorry, not the lysosome. That's one of the ways that your immune system protects you against bacteria and viruses, is by literally chasing them down in your blood and your tissues and getting rid of them. So yeah, this is happening all the time in your body. Isn't that crazy? It's weird, right? But also that's how your immune system works. So kind of cool. All right. So that's the endomembrane system. We've got the rough endoplasmic reticulum and the smooth endoplasmic reticulum. Rough and ER makes proteins. Smooth ER makes fats. They hand things off to the Golgi apparatus, which packages, sorts, modifies, puts it in a new vesicle, puts a label on it, and sends it off to where it needs to go. One of the things that the Golgi apparatus makes is a lysosome. Okay, in the next section, we're going to talk about the mitochondria. Yeah, they are the powerhouse of the cell.